Hola a todos, muy buenos días, bienvenidos a otra sesión de seminario del Departamento de Biología. I will be speaking in English so that Nelson and us, we decided this was the best language that everybody could communicate better. It is really an honor for me to have Nelson Vaz with us. He's a longtime friend of Maturana and the Rayo Labs of Jorge Podosis and Juan Carlos de Tellier. And through them, I have met Nelson. Although we've spoken most about systemic biology, he is a world authority on the immune system, which is a subject that, like many subjects in biology, is metaphor heavy, right? I would say that there's lots of language that is kind of metaphorical, kind of cognitive, but this is very interesting. Nelson has a more structuralist approach, a very fascinating approach to the immune system, practically as an ecosystem, as what I can remember. We've also a system that that learns, of course, that has a history, let's say, that we can explain in very interesting experimental approaches to that. Nelson has pioneered how diet can affect the immune system. So this is actually, it's always fascinating to listen to Nelson. A big friend, as I said, of Maturana, of the lab. So it's really an honor for me. And please, Nelson, tell us about the immune system. Thank you very much.
Well, I have a new share. Can you see this?
Yes.
Okay. So thank you very much for inviting me. It's an honor to be in the laboratory in which I have been so many times before. And this is an old affair. And I have just listened to Podosis and Juan Carlos talk about the involvement of Maturana with teaching, with learning. And immunology seems to be a pretty good subject to approach these problems, because for most people, for the public, and for most professional scientists also, including immunologists, vaccination is taken to be a biochemical command to which the body obeys and changes in a desired direction. So you're exposed to the COVID virus and you become more resistant because we decided this. We forced the organism to change in a defined direction. Well, this is wrong in many ways. So perhaps after the seminar we can have questions and understand the relation between learning and immunology in another light. So Podosis also said that many scientists tell him that their lives changed when they met Maturana. And my life certainly changed when I met Chilean neurobiologists. But the first one was Francisco Varela, not Maturana. And this created very peculiar problems for me. I'm sending already this list of publications that Alex will make available to all of you later on, but this is the background, the theoretical background of what I'll be saying here. But I'll be also go through experimental findings, especially more recent findings from the laboratory of my wife, Claudia Carvalho. So in addition to these 3 Chilean neurobiologists, I will be telling her experimental findings that I think are very, very, very relevant to this understanding. Well, I met Varela in Denver in 1978, and he was interested in the first systemic theory, the first network theory to explain immunological phenomena that was proposed by Niels Jerne in 74. Basically, it's a theory based in the existence of antibodies. I mean, antibodies are also ancients. I mean, if you make an antibody, your organism will be making antibodies that react with this antibody as well. So this makes a big network, multi-connected network. And this was the first theory approaching the genuine notion of a system in immunology. And we published a paper in which Francisco added to Jörner's theory the idea of operational closure. Well, this was 78, so this was after De Máquinas e Seres Vivos, the first book he published with Maturana, in which these concepts of closure and organization and structure were already quite well defined. So I was not aware of this. So I wasn't aware of Maturana. I never heard Maturana's name. Varela never mentioned Maturana in the semester he spent in the laboratory with me. So what happens is that I met him in a moment in which my laboratory was really undergoing a profound transformation, because we had stumbled in a phenomenon that also changed my life, because I worked with it for the next 40 years, which is immunology is called autotolerance. I will show you what autotolerance is, but the point is that Varela was not at all interested in autotolerance, as I was. So this is what we call autotolerance. If an animal eats or drinks a new protein like ovalbumin, hence ovalbumin is not a common protein in mouse diet, and if a few days later you immunize the mouse with the protein, the small amount of this protein, what happens is that the amount of antibodies that animals that drink water form are much larger than the amounts of antibodies that this animal that drank the protein before forms. This is autotolerance. It's not a small difference. Here's in radioimmunoassays, a control mouse, let's say, with this value of antigen binding in the serum. If you inject 2 milligrams of the protein intravenously, it also has a decrease, goes to a third of a naive mouse. But if he eats 20 milligrams of the same protein, which is very little in terms of food materials, this less than 0.1% of this material will go into the organism, but the effect is incredibly strong. So I was flabbergasted with this finding, because every animal eats every day hundreds of different proteins. If this happens with all of them, then this should be in the first page of the immunology test books, and sometimes it's not even mentioned. So then I began a very different track of research, and by our means also, we made a symposium in Brazil, to which we invited Maturana, Varela, and Antônio Coutinho, which already at the time was a world-famous immunologist. And Varela met Coutinho at this time and developed a friendship. And during about 10 years, he went to the Institute Pasteur, and they published a dozen papers on cognitive networks, also with John Stewart trying to make computer modeling of these experiments. In the unit was called immunobiology. For me, the trajectory was quite different, because I went from Rio, where I was a teacher, to Belo Horizonte in the mountains north of Rio, and made a new laboratory called immunobiology. And I had many students there, but these four ones I will be mentioning in this talk and made very significant findings in oral tolerance. The stay in the Institute Pasteur, I was also there in 1986, 87. Alfred Tauber is an important name in immunology, because he's also a philosopher. So he has this double view and has many books about the theoretical aspects of immunology. And in one of these books, and in the Stanford Encyclopedia, he creates this term, the Parish School of Theoretical Immunology, which includes Coutinho, Varela, and Stewart, and also my name. Well, actually, my name is here because of this paper, and I was included as an author, but I didn't participate in writing this paper. So, and also I don't agree much with what is said there. So I'm not, because of my interest in oral tolerance, I'm not to be included in the Parish School of Theoretical Immunology. So I'm really referring to three groups of interest. This group that is the genial neurobiologists, the group in my laboratory that was involved in oral tolerance, and the group at the Institute Pasteur with Coutinho and members that developed the idea of reliable patterns of immune responsiveness, blottings. And John Stewart was also a very important member of this group. So there are three different sets of ideas that interlock. When I came back from Denver, where I met Varela, I had changed my way of seeing the immune system, but this was before meeting Maturana. And already I was using the idea of operational closure, because this is somehow the classical, the traditional idea of how the immune system works. There are units, independent units, that may be clones or sets of clones of lymphocyte clones, and the anti-A will respond to A and the anti-B to B and C. They are independent from each other. And after spending a semester with Varela, my understanding was that suppose that anti-A clones have components that we call B, which are recognized by the set anti-B, and this also happens with the set B and C. So there is a circular organization, and the system, this is a stimulus that will produce a response here, but here it's more likely understood as a perturbation or an intrusion of a foreign element into something that has already a physiological, something that is already ongoing. Then I met Maturana. And the idea of the epistemological and ontological problems of cognitive systems. And he summarizes the problem about immunology in this sentence, because if we see the immune system as a cognitive system, we'll seek to explain and understand it in cognitive terms, according to what we think is a cognitive phenomenon. And generally, immunologists don't think very much about cognitive phenomena. I mean, there's this immunology is totally far from cognitive sciences, cybernetics, or anything like this. So I told this is a long story, because this is 1993. This is Maturana Pedrosa's Claudia, my wife. This is Pedrosa's wife at the time. I brought him to São Paulo for a course in biology of cognition. And it was very well succeeded. It was the first time I had Maturana to talk to a large audience in Brazil. But this was repeated many times after that. Well, at the same year, I published a book called The Incomplete Guide of Immunobiology, and Maturana wrote a preface to this book. And here is some sentences from the preface. That told me that he understood quite well what I was driving at, like the internal space becomes a closed molecular and cellular relational dynamic. Because for immunologists, the organism is simply the space or the dimension where immune responses take place. I mean, the organism is given. There's no attempt to explain what the organism is. And now, here, Maturana is saying that what we propose is that the internal space defines the unity of the organism rather than being defined by it. So it's a part of the construction and maintenance of the organism, or the vertebrate organism, is something quite different. So here again, the same point, it no longer has a spatial connotation and recover its dynamic relational character. The internal space, the organism is not empty. I mean, immunological activity is a continuous, a ceaseless, spontaneous, and non-anticipatory activity. It's quite different from immune responsiveness of traditional immunologists. Well, this went on for many years, and this is one of the last publications we made in 2017. With Luis Andrade, we spoke of the epigenetic immune network. And Maturana has a participation in this, made a comment on this article, a one-page comment, and he characterizes diseases as loss of inner harmonies. Well, if disease is a loss of harmony in the sense of musical harmony, right? What is a sonata? What's a symphony? There are rules for a sonata, rules for a symphony. But harmony, it's a very old idea. It was a very, very popular idea in 1850 or before 1850, medicine was still in the humors of Galenic medicine. So the organism had an equilibrium, an internal equilibrium, a harmony, and disease was a disequilibrium between these humors, the bile, the black bile, the yellow bile, phlegma. And all this was completely destroyed by the germ theory of Louis Pasteur. So when he says every disease has a little seed, a microbial seed, a specific phenomena caused by a specific microbe, and you can prevent these diseases by using vaccines, so weakening or attenuating the germs in laboratory and use the attenuated germs as vaccines. He had a totally wrong idea about the way vaccines function. He thought he had a nutritional explanation, said, well, you imagine he was discovering microbes for the first time, and culturing them in pure cultures. So every microbe has very specific requisites for nutrients. So sometimes microelements. And his idea was that the vaccine, the microbe would starve, would completely use a microelement in the organism. So the organism would be wrong terrain for this kind of germs, because they wouldn't find this microelement necessary for survival. But then people were finding vaccines worked also with dead germs. So his idea was blown up. But look, the germ theory that diseases are caused by germs and not by disequilibriums of the harmony was not simply accepted. Claude Bernard was a giant in science, the creator of physiology and a great part of experimental medicine. He said that what the important thing was the conservation of the internal medium. So if the internal medium was preserved, contagion would not lead to disease. So this is totally against the germ theory. Metchnikoff, which was working in Pasteur's laboratory, had a different idea. For Bernard, there was an internal harmony. And for Metchnikoff, the organism was in total disharmony. And in a big struggle, in a constant struggle to reach harmony. So this is to say that this idea of harmony is a very hot idea and a very old one. So when Maturana brings the idea that diseases are loss of harmony in the body, he's bringing back another angle of the debate. I heard from him that the real problem is this: what happens to the observer in his or her observing? We all know we have blind spots in the retina, for instance. And in the laboratory, you can put things in this blind spot which you don't see them. But if I lose my pen on top of my table, and I'm looking for my pen, and I look for it for several seconds, and then, here it is. Before that, the image of the pen was in my retina the same way. So the question is, what happened in this particular time where I make the distinction of the pen? What happened to the observer there? So, and his treatment of languaging, the peculiar way of living of human beings, he has, in his first papers, at least in his conferences in Brazil, he used to draw an eye in every graph. He'd drawn a phenomenon, but in every graph there was a little eye in the corner saying that there's an observer of what's going on, and this is what happens. This is said by somebody to somebody else. Well, then came Podoses. Well, I met Podoses much earlier. I met Podoses in 1990 in your laboratory. But he came to Brazil also several times. And this is Gustavo Ramos, which was very important in bringing us together, and in Santa Catarina to made several meetings there. And he's now in Germany, and he practically inaugurated a new kind of immunology of the heart. I mean, he thinks that official science will never understand what we say, because the problem that concerns us is an epistemological problem, but it's very subtle. the difference is very delicate and is something fundamental, yet very subtle. And I don't think there are many possibilities of official science might one day take into account. Well, but we have been working on this. We published a little book together, which is called "Where is the Organism?" And Podós, in one of his sentences, he says some things that are really very synthetic. And this one, he says there is plasticity in biological development. The ways of development are plastic at all moments. And this is what allows this wonderful diversity of lineages of living beings. But the problem is not what is plastic, but rather what is conserved. And here's the sentence. If change is a constitutive condition of living, then how is it conserved, that which is conserved? So if living is a way of changing, what's conserved? Well, Maturana would tell you what's conserved is autopoietic organization and congruence with the medium. But I think Podós is wanting something more specific than that. So in all these conversations with these three men, and also with Antonio Coutinho, with whom I learned a lot of immunology. Coutinho, I don't know why he didn't get the Nobel Prize in immunology, because he really is one of the most important European leaders in immunology. So our view of the immune system is that it's a part of the organism, or the vertebrate organism, like the nervous system. Both systems interacts and they interact with the organism. So the medium of the immune system is the organism. Is not the medium of the organism. The medium in which the organism lives is a meta-medium for the immune system. It never reaches the external medium. So our inner medium doesn't know of an external world. And our way of moving in the external world doesn't know of the internal organ, the internal world. I don't know how I talk. I don't know where the words I say come from. I don't know where they come from. So let me tell you something more then about oral tolerance, which is the strong experimental part of our postulation. This began with Donald Hanson in Denver, because I wanted to know if a mouse is able to avoid drinking a solution that has an enzyme to which it is allergic. So he had a PhD on drinking mechanisms of thirst, and we did the experiment, and then it's a wonderful result that gave other PhDs after this. Because if a normal mouse, if you put ovalbumin with a little saccharin in there, it will drink like 95% of the intake. If you put the 2 bottles, water and sweeten it over, it will drink like 95% here. And in a mouse that have been immunized to over, it's exactly the opposite. They avoid drinking this. So in the course of doing these experiments, Don came to me and said, look, Nelson, the mice that drink ovalbumin cannot make anti-over antibodies any longer. I said, you did something wrong, repeated, he repeated, and the result was very clear. There's a massive blocking of immune response maps to materials that are ingested as food. So this is what I already told you before. It's a very marked decrease in antibody formation. numerically, here it is. And here, the situation becomes more interesting because we found that we could give ovalbumin to a mouse, make it tolerant, so it will make little antibodies. It's blocked in making antibodies. So I get this mouse and I take T lymphocytes. T lymphocytes are the first complication, major complications in immunology. It's necessary to form antibodies, but it doesn't form antibodies themselves. They are helper cells. But if you transfer T lymphocytes of a tolerant mouse to a normal mouse that drink water, so you put these lymphocytes into this mouse that drink water, and then immunize it with ovalbumin, it will respond like a tolerant mouse. So this is an active phenomenon. Depends on the lymphocytes, depends on some organization of the lymphocytes. It took us 15 years to understand that even this is not an inhibition, because no tolerance is complete. But if you take this residual responsiveness after having received a large dose of an engine, now was not ovalbumin, it's lysozyme. A large dose of engine will make a response of normal mice, or mice that didn't drink lysozyme 3,000, he has 500. So if this animal receives several different immunizations with lysozyme, it doesn't change the level of antibodies in the blood. So you may gradate the level of responsiveness of a certain engine. So by the second exposure, the animal will lock its level of specific responsiveness to that antigen. So this is important because this is a new phenomenon in immunology. It's not a response, it's not an inhibition of a response, it's a conservative phenomenon. It's the establishment of a pattern inside the organism. This led Claudia to make her PhD with the attempt to immunize with tolerated antigens. So you know that tolerance has to do with the lymphocytes. You can transfer it with lymphocytes. So what happens if you inject the antigen to which these lymphocytes are responding? What happens? And she said, well, how can I tell that? I thought, well, try to initiate another immune response together. So we inject two antigens in the animal, one of them to which the animal is tolerant, the other not. And she found that the animal will not respond to the second antigen. Well, this led to a flurry of different experiments, but this is one of the most nicest one. These are granulomas in the lung of mice, of normal mice, injected with living eggs of Schistosoma mansoni worm. These eggs are big, and when they pass through the lung, they embolize in the vessels within the lung. They don't know they are in the lung. They continue to secrete their enzymes, and a granuloma is formed around them. Now, what she did was she injected the eggs in the animals, that at the same time, she injected a small dose of ovalbumin in animals that were tolerant to ovalbumin. So they had drank ovalbumin before, and these animals did not drink ovalbumin before. And the size of the granulomas are extremely strongly affected by this small amount of a protein that has nothing to do with the worm, with the eggs. This happens because there is an inhibition of acute inflammation caused by injecting the antigen. And this was done by testing in over-tolerant mice in the response to carrageenan. Carrageenan is a substance used by the drug companies to test anti-inflammatory substance, new substance. It's the standard test. So if you inject ovalbumin into an ovalbumin mouse, it will react less to carrageenan. So it's this definite anti-inflammatory phenomenon by the most popular test in the drug industry. So inflammation is involved in healing of skin wounds, for instance. So if you take a mouse that drank ovalbumin and make a cut with a razor blade in his back skin, dorsal skin, and at the same time you inject 10 micrograms of ovalbumin elsewhere and subcutaneously in the mouse, the scar is quite different. It's a much smaller scar. It's formed faster, and there is a host of cellular factors that Claudia and her colleagues and her students investigated that are changed by this injection of the tolerated engine. Now, among these effects, one of the most impressive is in the deposition of collagen fibers, because normal collagen in normal skin, they look like little baskets like that. And in scars, the collagen is linearly arranged. And that's why scars are weaker than normal skin to traction. So when you inject ovalbumin and you look to the collagen that's being deposited in the scar, it looks like normal skin collagen and not like scar collagen. So it's a morphogenic effect. And Gustavo Ramos did this in the heart. He injected isoproterenol, an overdose of isoproterenol into rats. So this kills, let's say, half the animals, but half survive this aggression. And it's like a myocardial infarction. He's going to die like 10 or 20 days later with heart failure. And if you induce, this is fibrosis in the heart of control animals, of injected with isoproterenol, and injected with ovalbumin and isoproterenol. It's almost like it's very close to the control. So you can change the fibrosis in the heart, in the ischemic heart, by giving the animal, exposing the animal to an enzyme to which it's tolerant. Which are the enzymes that the animal is tolerant? All the enzymes he ate before as food. Now, also the idea that tolerance is an inhibition of lymphocyte activation is not true. There are different kinds of lymphocytes activated when you inject a tolerated enzyme into an animal. So these are too technical to discuss now. Well, this make me able to make very general statements about the present understanding of immunology. I think immunology has been thrown in a misleading direction by two exceptional findings. one vaccine and one serum therapy, artificial serum therapy of diphtheria. Vaccines were invented much before antibodies were known. And smallpox was the most dangerous infections that the human race has faced. COVID-19 has killed, I think, about 6 million people up to this day. The calculus is that smallpox killed or maimed about 600 million people during 2 or 3 centuries. So the vaccine was a blessing, but it was an exceptional vaccine, and very exceptional for several reasons. There was no subclinical infections. The victims were infectious for 5 days in which they were in bed. The virus was completely eliminated from the body upon recovery. There was no animal reservoirs. The symptoms were clearly recognizable. Infected cases could readily be identified. And then an inexpensive, effective, and easily transported vaccine was available. And a planet-wide surveillance organization is maintained up to this day. There's no other disease that fulfills these items. The second model of immunological activity is Diphtheria Antitoxin. So when they start studying bacteria, they found that some bacteria like diphtheria and tetanus secrete very poisonous substance, toxins, to the cultural medium. So if you inject these toxins, you get the disease in the absence of the germ. The toxin causes the disease. So they also found that animals that survived the toxin became progressively more resistant. They searched for the source of this resistance was something in the blood, an antibody, an antitoxin. And then they also find that they could take serum from animals containing antitoxin and put into normal animals. They were protected. And they use this in humans. So children dying from diphtheria were treated with serum from hyper-vaccinated large animals like horses or oxes. And the conclusion was very clear. Vaccines probably make antitoxins to every bug, every virus. And that's how vaccines function. And this is not really not true, except in a very, very few cases. So in COVID-19, the conclusions are that I can tell you now is acute infectious diseases like COVID-19, they are human cultural creations. They are not the results of chance collision with infectious antigens. Let me jump here to the next. Immunology is generally the teaching of immunology starts with Pasteur, with the discovery of antibodies. But if you go into deep time, there is evidence now in the Neolithic period, between 10 and 6,000 years ago, devastating epidemics that was threatening to end human life at all in the Middle East. This coincided to, I was very impressed with this book, Against the Grain. Against the Grain is an expression if you go against the direction of the hair or the grain in the wood. But he's talking about against the cereals. He tells how the deep history of the earlier states where cereals, they are easily collected, stored, transported. The day of collection is previsible. So according to Scott, this was what created the tax collector and had a very important effect on civilization. But the point I want to raise is that this also is the time where mankind domesticated several birds and mammals that start to live in close proximity to humans. And this triggered this passage of these viruses, of these animals to humans. And it really caused the epidemics, which were then became endemic in the population that is spread over Europe. And then when the Europeans crossed the Atlantic and came to North America or Central America and to Brazil also, the same epidemics blew up again because these populations, the ancestor of these populations, did not live through the period in which agriculture was adopted in Europe and in Asia. So we may think that acute infectious diseases are accidental encounters with virus and bugs in the environment, and that's not true. I mean, plants and animals, without the influence of humans, of human culture, don't suffer acute epidemic diseases. There is now bird flu in America killing many birds. Frogs are being destructed by a fungus. All this you can trace back to the origin of a human intervention. And now everything is mixed up with global heating and other factors that humans have introduced in our lives. So that's about it. I would love to have questions from you, but this is the idea we have about immunology, and this was strongly influenced by my contact with people from your laboratory. Thank you.
Thank you very much, Nelson. That was extremely interesting. I will open to questions now. If we have any questions, you can make the question in Spanish or Portuguese, and you can also send us questions on YouTube. We are watching the YouTube channel, so we will be watching for. You can.
How do I unshare this? I go back to.
I can. I think where it says share screen, it should say stop sharing. I think so.
Stop sharing. Okay.
Yes.
Okay.
There it is. Okay. Thank you, Nancy. So do we have questions?
What about autoimmune responses?
I would say that either you say that all diseases are autoimmune or none of them are, because lymphocytes are involved in all diseases. And the fact that there are many diseases that we label as autoimmune, like diabetes, for instance, type 1 diabetes, the ones that are truly autoimmune, that affect children. Type 1 diabetes is an inborn defect in a transcription factor. So in mice, you can show that the pancreas is damaged in the animals prone to diabetes before the formation of lymphocytes. So, the more we learn about the mechanisms of disease, we find that Yeah, this is the body tweet. This is the wrong idea.
One question.
Yeah.
In regards to your experiments showing the injection of lysozyme, that I found it truly amazing that these mice, by the way, that you injected growing amounts of lysozyme and they would not develop such an immune response to the lysozyme when they had been previously immunized and after drinking it. Do you have any comment on what would be, for example, upper respiratory diseases? Would that be a way in which many people will swallow phlegm or stuff like that? Would you think that we are inhibiting, or well, not inhibiting, but reducing our response to those antigens?
Yeah, well, I have an MD, but not practice medicine. But there's one thing that I recommend people that have rhinitis, chronic rhinitis. And then if you put a dish with butter in your beside your bed, and at night and in the morning, you put that in a glass and drink, because the allergens that are in the air will fall in the dish. And the point is that I want you to put those allergens into the digestive tract instead of getting it to. And also recommend is don't. I had adult people kneeling in front of me, thanking for getting rid of rhinitis that they had for decades.
I'm going to try that. I'm definitely going to try that.
Don't drink the whole glass in the first day.
Okay, I will definitely do that.
Yeah.
Nelson, so I'm very impressed for the oral tolerance experiment. It's really spectacular. It's really spectacular. And Alejandro kind of recently asked what I was wondering about applications. At the end of your talk, I was a bit I'm interested in this, that you're saying that there's no such thing as pandemics in nature, if I understood correctly, that they all are kind of produced by the cultural
Human action.
At the level of agriculture, something like that, if I understood correctly, when we have.
Yeah, so like rinderpest has killed wildebeest and zebras and so on, and devastatingly in Africa. They all came from cattle. So without human treating cattle, imprisoned cattle and artificially raised cattle, there's no rinderpest.
Could that be a density thing, that we keep these animals too close to each other, too many of them?
Yeah.
So that there is the transfer possible.
Yeah, and there's complications like this destruction of the amphibia is being boosted by the increase in the temperature with global heating. So this is human action in another dimension. So things get much more complicated. Invasive species that humans take from one place to another, like Hawaii. Hawaii is known as one of the points of the planet with a massive invasion of alien species that brought by humans. Australia with rabbits and so on. Humans have been messing up the planet and with these delicate ecological equilibria, but I believe that most diseases are chronic. Human diseases are chronic. We are not to underestimate what happens with worms, for instance, and fungi. But acute viral infectious diseases are human creations.
You mean that's in inadvertent human creations, not in a Wuhan lab kind of of way?
No. They are cultural creations. They are cultural creation. For the term now is transboarding. So it's the overflow of virus from one species to another in a zoonosis, passing from an animal to another animal. Now, bats, for instance, I used to think there were 3 or 4 species of bats. There are 1,300 species of bats. They are a nirvana for virus because they have an inflammatory mechanism that is quite mild. So they tolerate the presence of many viruses that no other animal will tolerate. Now, they fly, they disperse the virus to long distances. They are crowded and they are in touch with us. They feed each other. So they are optimal for the maintenance of. So if we start messing with ecological niches of bats, we'll be in big trouble very soon. Mm-hmm. And Brazil is certainly messing with everything in the Amazon. We're destroying the forests at a very high speed.
I have a question here on YouTube from Daniela Salma. She says, thanks for your talk. What about oral polio vaccine?
Polio vaccine is a enterovirus, so the virus, it colonizes the gut or spreading to the body. So it's basically an infection of the intestine. So it's not anymore an ancient, like you give albumin to drink or a protein like that. It's a virus that's going to colonize and have all kinds of effects in the gut. The gut is the organ of the body that has the largest number of activated lymphocytes. So the question has a very interesting side, which is why we don't have more oral vaccines. Say oral vaccines should be the future of vaccination. I mean, should be really in this direction. So if you want to put an antigen in contact with an antigen or with an activated lymphocyte, the way to do it is to drink it. Instead of, you reach more antibodies, more lymphocytes than putting it to the vein. So lymphocytes in the blood are in transit, resting lymphocytes, or very small amount of activated lymphocytes going somewhere else, and they can't do anything in the blood. There are new evidence that, for instance, the interlinking of collagen fibers in the body changes during aging. And in an old body like mine, T lymphocytes have a very big trouble to travel through tissues. So one factor important in my immunodeficiency is that my lymphocytes cannot travel in the body as they did before because of the old collagen I have. So the situation, it's not linear. It's not an ancient producing body that kills a bacterium. That's absolutely out of question. It can't be understood like that. Pile.
Pile, we can't hear you.
Could it be that we have been over sanitized in our modern culture, that if we had been as children more prone to take in all sorts of antibodies just by the way we as children act? You know, I remember picking up flies from the windowsill and checking them out.
Yeah.
Does that possibly create a better resistance?
Sure. And I'm quite convinced that the greatest idea in the biomedical area in the 19th century was the germ theory. So the idea that there is a microbial world. This was fantastic for mankind. That this invisible world of microbes. Now, the main idea in the 20th century was to understand that this microbial world is thousands of fold larger, more diverse, and more ubiquitous than we thought, and that we live in a soup of germs. There are 700 different microbes characterized in human saliva. So the idea we have now that this is a microbial world, and you have to understand that the multicellular organisms, they appeared in a world that was already inhabited by the by the microbes, and tenfold more viruses. And a large proportion of our genomes are also of retroviral origin. So this is a different world, it's a different biology. And the literature is chock full of effects of the native microbiome or microbiota and the relations with the immune system and all kinds of manifestation of health. And living in artificially clean, poor environments certainly is not a very good, healthy way of living. And our idea about the sources of immune pathology, so diseases that involve the immune system, is in the direction of a loss of complexity. I mean, we have to learn to live with complexity and with unexpected changes. We are always improvising. Life is improvising. And we are not ready for this kind of understanding because we are trained to understand the world in terms of fixed things. I mean, this table won't move. And the soil won't, that door is fixed. I will find the same door tomorrow, and a door knob will be the same place. And I teach my children that the Earth's two movements, and there are planets, and the sun is moving, and so on. And when she's 10 years old, I will talk her about molecules and DNA and so on. And we need a different kind of panorama now.
Well, can you hear me? Yes, it seems like the internet is having problems. It's amazing that, so I gather it's your strong opinion that immunological research should have more directions than the ones that are currently favored. So what would you say? And yeah, this linear thinking, is that the main obstacle? Could you elaborate final to close the seminar? What do you think are the problems that are stopping research from going in more interesting directions, perhaps?
Yeah, it's a tragic situation because we have an enormous influence of the drug industry. Now more than ever, with the pandemics, the drug companies are now in a position they never have been before. a very important role in decisions like that. But the decision of the drug industry was to produce monoclonal antibodies. The antibodies are an example of more heterogeneous substances we know. I mean, there are hundreds of thousands antibodies. If you, let's say, if you eat peanuts and want to know how many different kinds of antibodies you form to pea proteins, thousands of biochemically different antibodies that bind to these proteins with different degrees of affinity and so on. And there are antibodies that bind to these proteins, anti-antibodies, and all kinds of network problems there. Now, drug industry devised a way of making monoclonal antibodies. So instead of 1,000 antibodies, you have only one clone. So, and it's not grown in animals anymore. It's growing cultured dishes. And it is very expensive, but the protein is very homogeneous. So if antibodies were what they think they are, monoclonal antibodies would be in all laboratories, in all techniques, making them much better. They're very rarely employed because they are not what we expected it to be. And they are used in medicine also to block this or that cytokine, inflammatory cytokine. And they work. Sometimes they work marvelously well. And in the next patient, they fail completely, and we don't understand why. Vaccines still invented by trial and error, the same way Pasteur was doing in the 19th century. We don't know how to do it. So you say yellow fever vaccine is one of the most efficient vaccines. You need one dose to get lifelong protection. It's a flavivirus. There are 3 other flavivirus that you want to vaccinate, like dengue, chikungunya, Zika virus. You do exactly the same as you do with the yellow fever virus. There's no protection. So the thing cannot be resolved linearly. I mean, it's a very complex, interconnected system. We have to understand more. Worse, system biology now has many meanings, some of them completely contradictory with the idea of this interconnectedness that I'm talking about. So that's why laboratories like yours is so important for understanding our understanding.
Thank you, Nelson. It's been a really neat talk, fascinating. So thank you very much. I excuse Dr. Podosas and Letelier. They couldn't be here, very unfortunately. Some people took their time slot, but they send their apologies. They will be watching this video thereafter by YouTube. So thanks again, and goodbye, everybody. We'll see you next Wednesday in another seminar. Bye-bye. Thank you, Nim.
Goodbye.
Gracias a todos.